So first of all, I want to thank Dan and as well as Suzanne and Peter for inviting me to this super fun and exciting event. And I did think about some things to say about Dan, but I want to actually save them for dinner when Peg and the kids are also there. So unless there's huge objections to that, that was my plan. All right. So as you all know, there's a number of diseases that arise from a disruption in protein homeostasis. And of course, these include cancer. These include, there we go. So of course, these include cancer, neurodegenerative diseases, but also such physiological processes as aging. Now, of course, protein homeostasis is broadly defined as the equilibrium between protein production and protein degradation. And because protein production happens on the ribosome in all cells, the ribosome is really a central player in the protein homeostasis network. And when it makes mistakes, which could arise either from the mistakes in decoding the sequence information that's contained in the mRNA, wrong proteins, of course, are produced. And these might be prone to misfolding and thereby disrupt the protein homeostasis network. Or alternatively, of course, ribosomes must also decode the information in the mRNA that specifies how much of any given protein is produced. And therefore, if it makes mistakes in interpreting this information, too much or too little of any protein could be produced, and this could also, of course, disrupt protein homeostasis. Now, as you might know, my lab has been interested for the last 12 years in trying to understand how ribosomes are produced. And it turns out that correctly assembling ribosomes has been a central interest of ours, and this is really important to maintain protein homeostasis, as we know from the large and growing number of diseases that arises from point mutations in this huge machinery that's required for assembling ribosome, or from haploinsufficiency of ribosomal proteins, which can also lead to different diseases. And I've only listed a small subset of those, which are also concentrated on the small subunit, because that's really what we've been most interested in. So what I want to tell you about today is that cells, under conditions either of disease or stress, manipulate the composition of their ribosomes to produce different ribosomes which have a different specificity or maybe more like selectivity to different mRNAs, which then can lead to the production of a different set of proteins. Now, as you all know, ribosomes are complex molecules. In all forms of life, they're made up of two subunits. And in eukaryotes, these subunits themselves are composed of 4 ribosomal RNAs and 79 ribosomal proteins. So of course, if I give each one of you guys a set of 80 Lego pieces, each one of you guys would build a different thing from it. However, there's only one way of constructing ribosomes correctly. And so it's a really tremendous challenge to put ribosomes together correctly. And as you can see, my nephew's also figured out different ways of putting these together. Now, moreover, the second challenge is to make sure that really all of the pieces are incorporated and there's not one that fell under the table. And of course, this is especially challenging for ribosomes because many of the polypeptides that are composing the subunits are really very small polypeptides, many of them under 10 kilodaltons, and it might be hard to find them in a molecular machine of several megadaltons. And this challenge is even further exacerbated because it turns out that the ribosomal proteins can bind ribosomal RNA in many parallel pathways. So there's a lot of different intermediates, all of them are on pathway, and this really makes it very challenging to quality control them because they're really all supposed to be there. So to understand really how cells ensure that all ribosomal proteins are incorporated, we decided to use as an example the formation of this protein network at the backside of the small subunit head. You can see there's a number of different proteins which are all centered around this red protein RPS3. And it turns out that there's a number of data points in the literature that already showed that this one specific assembly factor, LTV1, which bound directly to RPS3 as well as its RPS20, was somehow perhaps involved in their incorporation. The as they all bound to each other. So to dissect the role of LTB1 in the assembly of this protein network, what we started out with was a structure probing experiment. So what we did is we looked at the accessibility of the RNA either in wild-type yeast cells or in LTB1 deficient yeast cells. And you can see that there are a number of residues that are more or less accessible, which are quantified here on the bottom, and then mapped onto the structure of the small subunit here. And you can see, first of all, that they are clustered in this two sets of helices, and gratifyingly they were adjacent to RPS20 and RPS3. And I already told you those are the two proteins that bound directly to LTB1. So that makes sense. However, somewhat counterintuitively, they were kind of on the wrong side of this protein complex, because this is where LTB1 was thought to bind. Now, what I think is going on there became more clear when very recently Will and Beckman's lab published a structure of 4DS precursors. So what we are looking at here is basically at the small subunit head from the top down. You can see that there's a junction that is not yet formed in these 4DS precursors. And it turns out that this juncture is the exact juncture that is bound by RPS3 in mature ribosomes. And so that is exactly the juncture that we're looking at here. So what I think is happening is that LTB1 prevents the premature interaction of RPS3 with this RNA element, and therefore prevents somehow misfolding of this element. So next, what we wanted to ask then is does this also affect the incorporation or the positioning of RPS3 and the incorporation of all of these ribosomal proteins? And the first probe that we used to ask this question was really genetics. So as you can see, we have these structures of mature ribosomes. And so what we did is we introduced a number of point mutations within this protein network and asked how important are these point mutations in the presence and absence of LTB1. And so we first started with this interaction here between RPS3 in red and RPS17, which is really with this tail of RPS3. So basically we're asking about the positioning of this tail. And as you can see, these point mutations in RPS3 that interrupt this interaction on either side of the two proteins have small effects on growth, but these go entirely away in the absence of LTB1, leading to this epistatic effect that we're observing here. And so this is, of course, exactly what you would expect to happen if this tail is mispositioned in the LTB1 deficient cells. Now then, to ask about the position of these globular domains of LTB1, we made another set of point mutations either in RPS3 or RPS20 that disrupted these interactions, which are all highlighted here in space film. And you can see that generally these these point mutations become more deleterious in the absence of LTB1. So what this suggested to us is that these mutations did something that was exacerbated by the absence of LTB1. And considering that they were all in this protein interaction network, what we started to wonder was whether these really affected the binding of these ribosomal proteins, and that LTB1 was also affecting the binding, so that together it was more catastrophic effect. And so we used my quantitative Western blotting on purified ribosomes, either from wild-type or LTB1 cells, to show that indeed RPS10 is incorporated in a lower stoichiometry in these LTB1 deficient cells relative to wild-type cells. And it turns out that the same is also true for ASK1, which is bound here to the tail of RPS10. So to summarize what I've told you so far, as well as a number of other results that either I don't have the time to, well, I should have told you this here. I think I got distracted by the whole thing. So it turns out RPS10 and ASK1 are incorporated substoichiometrically. However, RPS20 and 29 seem to be incorporated just in the same way as in wild-type ribosome. Okay. So to put all of this together, what we think is going on is that LTB1 and its binding partner EMP1 help recruit these two ribosomal proteins very late in assembly, presumably by direct interaction between LTB1 and RPS20. And this then helps, LTB1 also helps in the recruitment of RPS3. Now what LTB1 is apparently doing is it's keeping RPS3 and RPS20 apart from each other so that they cannot form the interaction that they make in mature ribosomes. And it's also preventing thereby RPS3 from binding prematurely to the ribosomal RNA helix that I showed you at the beginning. Now, once this helix is formed, a kinase actually comes in to phosphorylate and then release LTB1. And it is only after the release of LTB1 that RPS10 and ASK1 then bind. And of course, this requires the proper positioning of RPS3, and it is this that's then also disrupted in the LTB1 deficient cells. So next, what we wanted to know is what effect has this, the presence now of the second population of ribosomes on translation in cells. And so the first way that we address this is by using a set of reporter plasmids that measure the fidelity of all steps of translation, the recognition of start codons, stop codons, as well as like a normal codon in the middle of the mRNA, and frame shifting. And you can see that there are small defects on translational fidelity that all involve decoding processes. And we think that they arise from the deficiency of RPS10 because it turns out that RPS10 deficiency produces very similar defects. Now, Susan Lundquist's lab a number of years ago has shown that stop codon read-through produces also resistance to certain stresses in yeast. And presumably this is because it stochastically expands the proteome. And so correspondingly, we also found that LTB1 deficient yeast are resistant to stress induced by caffeine, ethanol, or cadmium chloride, but not lithium chloride. Now, however, what we really wanted to know was whether this distinct set of ribosomes that was formed in the absence of LTB1 also had a different preference for different mRNAs. Now, I should say that Wendy Gilbert's lab has shown this already for ASK1 deficient yeast, and she has specifically shown that ASK1 deficient yeast have a hard time translating small mRNAs, and these are more effective than large mRNAs. And I should also say that we're still working out these questions for the example of RPS10. However, we have shown this for another ribosomal protein, RPS26, which actually binds even after these two proteins I've been telling you about. Now, I won't have the time to show you in detail, but what we have shown is by separating ribosomes that contain and lack RPS26 and sequencing the mRNAs bound to it, that RPS26 is involved in recognition of one of the residues in the Kozak sequence. And then also it turns out that the mRNAs that are affected by this differential recognition are not randomly distributed over the yeast genome. Instead, they cluster in specific pathways. So, and specifically, you can see that mRNAs that are preferentially bound by RPS26 deficient yeast, which are shown here in blue, are enriched in encoding for proteins in the HOG1 pathway, which is a MAP kinase pathway encoding the response to high salt stress, as well as the RIB101 pathway, which involves the response to high pH. Now, correspondingly, RPS26 deficient yeast are actually resistant to high salt and high pH stress. In fact, not only are they resistant, they grow faster when you throw them into high salt or high pH, and of course this is different from wild-type yeast. And most interestingly, yeast cells also make RPS26 deficient yeast when you put them into high salt or high pH, but not when you throw them into other salt stresses. And this is relative to a number of other ribosomal proteins, which is quantified here. So this is interesting. It shows for the first time that there are physiological roles for these ribosomes that lack individual ribosomal proteins. However, it involves yeast, and yeast are an interesting model and a useful model organism. But does it also have more important roles in human physiology? So what I'm showing you here is there are some data from the literature where plotted are a number of different cancers and the expression of all the different ribosomal proteins in these cancers. And what you can see, even without being able to read anything in particular, is that these ribosomal proteins are not uniformly up and down regulated in these cancer cells. In fact, you can see that in some cancers, some proteins are specifically down-regulated, and shown here in blue, and then in other cancers, other proteins are specifically up-regulated, which is shown in red. So in other words, cancer cells actually generate ribosomes with not normal stoichiometries, lacking individual proteins. So we wanted to know if this was also true for this mechanism about LTB1 that I was telling you about earlier. So when we first looked at the LTB1 levels in two cancer cell lines, either glioma cell lines or breast cancer cell lines, we didn't find a large difference in the levels of LTB1. But when we looked at the amount of ribosomes that were produced by these two cell lines, we noticed that breast cancer cells make almost make more ribosomes. So even though the levels of LTB1 are the same, they're not sufficient apparently to produce all the ribosomes in breast cancer cells relative to glioma cells. And as a result, it turns out that in breast cancer cells, the ribosomes have substoichiometric amounts of RPS10 and ASK1, which in human cells is called RAC1, as well as RPS3. And similar to the yeast cells, they also have these defects in stop codon recognition. So I've shown you already that these cells from LTB1 deficient yeast have resistance to certain stresses. And you might imagine that this could be one of the reasons why cancer cells produce these types of ribosomes. However, what we started to wonder is if they also might have unique vulnerabilities, and that's, of course, one thing that cancer researchers are specifically interested in. Now, and the thing that we were in particular interested in is kind of schematized here. So it turns out that there's a quality control, RNA quality control pathway known as the no-go pathway, which really recognizes stalled ribosomes. And these might be stalled because of defects either in the mRNA or the ribosome, and it is recognized through the same molecules. And it turns out that one of the required components on the ribosome for this pathway is known to be ASK1. So we predicted, apparently correctly, that LTB1 deficient ribosomes would also be deficient in this quality control pathway. And because in vivo, one of the main substrates for this pathway is presumably oxidized mRNA, we tested if these cells are then sensitive to oxidative stress, and as you can see that this is the case. So to summarize what I've told you, I've showed you that LTB1 is important as a chaperone for ensuring the correct folding of the ribosomal RNA, and therefore it's also important for correctly positioning RPS3 and then the subsequent recruitment of ASK1 and RPS10. And I've shown you that this role of LTB1 can be subverted in cancer cells and perhaps also under stress conditions to produce ribosomes that have a slightly misfolded head, which results in mispositioning of RPS3 and substoichiometric incorporation of ASK1 and RPS10. And I've also shown you that those types of ribosomes can have different translational properties, both in terms of fidelity, but also in terms of the mRNA selectivity. And last, I of course need to thank the people who did this work. So the work on LTB1 was really a collaboration between Jason Collins and Homer Galei in my lab, and they were helped by Joanne Dougherty, as well as Rebecca Culver, who was a summer undergraduate, and I'm mentioning this here because she's actually now a graduate student here. This is how the world is very small. And then the work on RPS26 was done by a graduate student in my lab, Max Ferretti, and he was helped by a rotating cast of high school students and undergraduates, including Ethan Ward, who is now at UCLA, Elizabeth Potts, who is now at the University of Florida, and Jennifer Barr, who is now at Georgetown. And I want to thank these people who pay the bills, and again, I mostly want to thank Dan for turning 60 and all for inviting me. Yeah, we haven't looked at this. There's no evidence for that. And then we would predict that there would be changes in the mRNA levels. Yeah, we haven't also really carefully looked for this. There's probably ways of doing that, yeah.
In the chaperone function of LTB1, how does the kinase know that The time is right.
Yeah, that's an excellent question that I can speculate. So we have been, it's a complicated network of events. So I've only been telling you about the proteins on the back side of this. So LTB1 binds to both RPS3 and RPS20. So there seems to be some unpublished data that was presented at a meeting that RPS20 is part of this network. And this network also then involves assembly factors on the front of the subunit. So RPS20 can communicate to the front. And so there, what I think is involved is, there is another kinase on the front of the molecule. However, this one is stably bound. It's there for a long time. And so what I think is actually happening is a complicated set of events. So we have data that suggests that the joining, so let me backpedal. A number of years ago, we've shown that it's actually the joining of large subunits to these 4DS molecules to produce what looks like ADS ribosomes that is part of a quality control mechanism. So we have evidence that it's the joining of EIF5B that's leading to the dissociation of the kinase on the front. We have structural data from ADS-like ribosomes that once this kinase on the front is gone, this juncture is formed. So I think it's the formation of the juncture after the front kinase dissociates, which is then somehow communicated to RPS20, which then somehow changes something for the kinase on the back in ways that we don't understand. And I figured this out in the airplane. It's a good thing I came. Yeah.
So you showed some examples of the RPS26 cells that can shift to higher growth rates, despite being deficient in an assembly factor. Mm-hmm. How are you thinking about that? I would think that deficiency would slow down by then.
Okay, so it's all relative. They're growing very slowly, but they're growing faster in high salt relative to how they are growing in low salt. But they're not growing, and at high salt, they're actually growing the same as wild type. Yeah. Mm-hmm.
Do you have a sense for the mechanism whereby specific mRNAs are selected or enriched by the LTB1 deficient ribosomes? And or have you also looked at, do they produce alternative peptides either by mass spec or some other mechanism?
So we haven't yet done, so we haven't looked at mass spec or anything like this. As I said, we are trying to look right now at the RPS10 deficient ribosomes. And really, as I said, the LTB1 is a combination of the two. So I haven't decided if I think it's better to look at to deconvolute, to start it in a deconvoluted way or in a convoluted way. So we might in the end have to do the convoluted way because the RPS10 is turning out to be a real pain. Anyways, that's just a sidetrack. So I think actually that there are two different ways that they're going to be having effects. One of them I think is really, it's kind of interesting, but in a boring kind of way. So I think that there are effects from low ribosome numbers on all steps of translation. So there's theoretical work that explains it on mRNA selectivity. I think that there are also effects on all of these other fidelity steps that we're testing right now to explain how non-second order steps could have effects. But I think I have a model for it. It's a very circuitous model, so we're testing that. And then RPS10, we have some data also that, so RPS10 is, so when you, if this is the small ribosomal subunit, the mRNA will bind here. RPS26 would be here, and so it's binding the Kozak sequence. RPS10 then goes through kind of like this little hole, then the mRNA goes through a hole, and it'll find probably RPS10 on the backside. I'm saying probably because all of the structures, the mRNA pieces aren't long enough to know. They don't comprise the entire footprint that we know to be there from all of the ribosomal footprinting experiment. So probably RPS10 is contacting the mRNA on the back, and we have some data from reporter assays that would support that being the case. And that's a lot harder to think about because these then are sequences that are already in the open reading frame, right? But we also, I've only been talking about how these interactions could affect translation initiation, right? Presumably the mRNA, for all we know, the mRNA makes contacts with the ribosome throughout the translation cycle, and these are identical to the contacts at initiation, which is all of the structures that we have, right? Actually, there's some structures about, for termination codon. So these interactions could also perturb the efficiency of termination, right? They could also affect stopping, right, which is what you were asking, right? And there's evidence from bacteria that Shine-Dalgarno-like sequences in the open reading frame can lead to stopping. And at some point, when you make the stop long enough, then it looks like a stalled ribosome, right? So there's a lot. I think it's a little bit overwhelming to think about. I think it's time to just do an experiment. And,
Thank you.